So hello everyone and welcome to our webinar dedicated to cleaning and disinfection in the agri-food industry. So today we would like to give you a case study of COVID-19 contamination. So the speakers for this presentation today are Lysianne, Ernst and I let Lysianne to introduce herself.
Yeah, hello everybody, my name is Lysianne Ernst. I have a scientific background as a biologist and I work by Rialco as an enzymatic application specialist.
And I am Laurent Delhalle and I have a PhD in microbiology and especially in risk analysis in the food chain, and I work for Rialco as a project manager. So before we begin, we need to focus on the objective of this presentation because the subject is very broad. But the purpose of this presentation is to give the keys for a good cleaning and disinfection protocol applied to the specific risk COVID-19. If you would like further information on how to deal with this risk COVID-19, including staff measures, we invite you to look at the document published by the World Health Organization. So for the agenda today, this presentation is divided into questions. We think that it's also very important to give some basic notion. For example, which molecule to choose in function of soiling or the activity of the risk. So we will do a little reminder on viruses to further develop on COVID-19, and we'll finish with regulatory aspects. So for the first question, I let Lysianne to explain which molecule to choose for cleaning and disinfection. So Lysianne, the floor is yours.
Yeah, thank you, Laurent. So first to introduce, I will just introduce what's hygiene. So hygiene ensures you to have and to work in a clean environment. It's based on your hygienic plan, your will, and your common sense. But why hygiene is so important? So it will guarantee you the quality of the finished product, and this will also guarantee you the food safety of your consumer. Now another question is, why do we have to clean? So cleaning is more or less obvious. You will not work in a dirt environment. But now how do we have, or why do we have to disinfect? Disinfection will control the microbiological risks in your work environment. This leads me to the definition of a detergent. So the detergent will remove the inorganic and organic residues of your surfaces. We will achieve what we call the visible clean. You will really see the results of a detergent. There exist a lot of different detergent classed by acidic product, alkaline product, or neutral product. On the other hand, a disinfectant will destroy and reduce the microorganism of your surfaces. So here we will act on the invisible clean. You cannot see microorganisms on the surfaces. And here we will also have different types of biocides. I will explain in some minutes. Let's start first with the different detergents. So first of all, the acidic detergents. So acidic products will permit to discard the surfaces. You have to know that you have to know that discarding is really important in your protocol because a slime film or a light film of scald on the surfaces can permit a point of attachment and protection to the microorganism, and therefore they will allow to grow. On the other hand, alkaline detergents, mostly high pH products, so you have to require precaution for the operators and the equipments. Most of alkaline detergents are composed by different wetting agents, also called surfactants, and the action is that they peel off the dirt of the surfaces, and then you can remove it by rinsing. Most of alkaline detergents will also have an action of cooked and carbonized organic residue. In revanche, the neutral or enzymatic detergents have a neutral pH, thus they are less aggressive. And every type of organic residue is handled by a specific enzyme. So you can see on the picture there exist a lot of proteases who act on proteins and meats, lipases who act on lipids, amylases on starch, and finally cellulase who will act on cellulose or vegetable fibers. And most of enzymatic and neutral products work with an enzymatic cocktail. That means that we mix a lot of different enzymatic types, a lot of different proteases, lipases, or amylases in a product in the aim to reach a deep cleaning. So this is to finish the part or the small overview of the detergent, and now we will pass on the disinfectant. So just to remember, a disinfectant will reduce and destroy microorganisms of the surfaces, act on the invisible clean. In relation of the sector, there exist different biocides. So in the healthcare, we will mostly see hydrogen peroxide, alcohols, sodium hypochlorites. In the food industry, we will more see chlorine products, quaternary ammonium, hydrogen peroxide, alcohol, and so on. But now, how do we have to choose the right molecule, the right disinfectant? First of all, you have to see, or you have to know that they have to respect the regulation of your country. Then you have to choose the molecule in function of your activity, the risk, the efficiency you want to reach, the equipment and the environment in which you work, the safety for your user, and the expected hygiene level you want to reach. Just to notice that for the efficiency, you have to compare and you have to know the different standards and norms to which the biocides respect. For example, a biocide can be bactericidal but not virucidal, so take care about this. Here in this table, you will see a lot of different sanitizers. So we will have chlorine products, iodine products, quaternary ammonium, aldehydes, hydrogen peroxide, peracetic acid, alcohol, amine, and so on. And on the right side of the table, you will see the different types of microorganisms. There exist bacteria who can be gram-negative or gram-positive, viruses, fungi, spores. And to choose the right molecule, you have to know the different advantages and disadvantages of a product. For example, chlorine products are really aggressive, so you have to use it with precaution, compared to the quaternary ammonium, who are less aggressive and therefore safer for the equipment and for the user. However, you have to know that chlorine product has that we call large spectrum. So they will act on different types. So the bacteria, gram-positive or gram-negative, viruses, fungi, and spores. In revenge, quaternary ammonium will have a smaller spectrum. So they will not act on every type of microorganism. And these analyses have to be done on the different types and on different biocide agents. So before we will finish the third part, I will just talk about the two-in-one solution. I don't know if you know something about the two-in-one solution. These are detergents who contains biocide agents. So you will clean and disinfect with only one product. What are the positive points of this two-in-one products? So first of all, you will gain time because you just have to apply one product and not two. Therefore, you will reduce the risk of interference with other substances. So you will avoid that the disinfectants will be inhibited by some detergent residues. You will save water. You will also eliminate the risk that the operators just want rapidly to disinfect a dirty surfaces or a dirty environment. And you will also avoid the product confusion. So you will avoid that people are cleaning with a disinfectant and then disinfect with a detergent. So to really resume to what we talked about, we saw that cleaning as well as disinfection is really important. You have to choose and to select the right molecules and active agents and to choose the right cleaning methods. You have to consider different factors such as the activity, the type of soil and the type of biological risk you will have, the material you have to clean, and the operators. And finally, you have to check the efficiency of your protocols. But if you want to have more information about how to clean, how to have a good protocol, I invite you to register to the next webinar, who is the following week. So this will finish the first pass, and now I will let Laurent talk you about viruses.
So thank you, Lysianne. And so we have seen that there are several parameters to choose the good cleaning and disinfectant product. So it's not an easy way to choose the right molecules, and how to choose the right molecule for COVID-19. So let's go back to viruses with some basic notion about virus and how to classify them. So virus is an infectious agent of small size and simple composition. They can multiply only in living cells of animals, plants, or bacteria. And the name virus is coming from a Latin word meaning poison. Viruses should not even be considered as an organism because they are not free living. They cannot reproduce and carry on metabolic processes without a host cell. So how to describe a virus? Briefly, a virus is made up of genetic materials housed inside a protein shell or capsid. The genetic material or genome may consist of single-stranded or double-stranded DNA or RNA and may be linear or circular. Viruses could be also composed of a lipid membrane surrounding the capsid, and they are called enveloped virus. So the criteria used for classifying virus are primarily based on three structural considerations. One, the type and the size of a nucleic acid. Two, the shape and the size of the capsid. And three, the presence of lipid membrane. So in this diagram, it gives the structure of non-enveloped and enveloped viruses. And enveloped viruses are sensitive to detergent due to the presence of lipids. These lipids are degraded by solubilization, leading to the inactivation of a virus with specific molecules such as detergent or alcohol. So in this graph, we have a classification of virus. We have DNA virus and RNA virus. And we have also a second classification with enveloped and non-enveloped virus. And we can see that the coronavirus or COVID-19 is an RNA virus with a lipidic membrane. So more specifically for COVID-19, the coronavirus causes the severe acute respiratory syndrome, the SARS syndrome, and spread quickly among humans. SARS-CoV-2 compared to SARS-CoV-1, SARS-CoV-2 is the COVID-19, cause significantly greater mortality, particularly among humans over age 65. At the beginning, some people compare COVID-19 to the flu, but here we have some key facts and some key values about COVID-19 and the flu. So we can see that the hospitalization rate and the mortality rate, it's much higher in the case of COVID-19 compared to the flu. In the right side of this slide, we have a structure of COVID-19. And the virus is nevertheless fragile and sensitive to detergent and alcoholic solution due to its lipid membrane. And the virus can be effectively inactivated by common disinfectants, including ethanol, chlorine, peracetic acid, etc. So let's go further with COVID-19. Is it present on surface? Is it present in biofilms? So current information indicates that COVID-19 is transmitted first during close contact through respiratory droplets from person to person, and secondly, on contaminated objects and surfaces surrounding the infected person. So recent research evaluated the survival of COVID-19 on different surfaces and reported that the virus can remain viable up to 72 hours on plastic and stainless steel, up to 4 hours on copper, and up to 24 hours on cardboard. So following the World Health Organization, it is imperative for the food industry to reinforce personal hygiene measures, to provide training on food hygiene principles, to eliminate and to reduce the risk of food surfaces and food packaging materials. So in this context, food industry is strongly advised to provide personal protective equipment, to introduce physical distancing, and to use stringent hygiene and sanitation measures by choosing, for example, the good molecules and the good cleaning disinfection process. So, second question is COVID-19 is present on biofilms. So there are few literature dealing with human pathogen viruses in biofilms, but there is evidence that biofilms serve as an absorbent and protective zone for viruses. Viral absorption on surface is multiparametric and is dependent on the physical properties of the medium, the surface, and the virus itself. So various studies have suggested that biofilm may trap and accumulate virus particles and produce a potential reservoir of human and bacterial pathogens. So, for example, viruses present in the biofilm are 30 times more resistant than viruses present in water during chlorination treatment. So inside the polymeric matrix, the virus can take advantage of a specific biofilm lifestyle, and in particular, the protection against environmental stress, such as desiccation and the presence of antimicrobial agents. So what is the relevance in a public health point of view? So biofilm were already regarded as a common cause of bacterial infection, but now they should also be considered as a protective reservoir of pathogenic virus and are probably responsible of numerous persistent viral infections. So now, how to eliminate effectively COVID-19? And I'll let the place to my colleague Lysianne. So Lysianne, the floor is yours.
Yeah, thank you, Laurent. So Laurent told us just that COVID-19 is an enveloped virus, and how you can see it here on the picture, the enveloped viruses are less resistant to biocides. Considering the different biocide agents, or if you will act on viruses, we talk about virucide agents, we know that they act on different parts of the viruses. So just to remember, an unenveloped virus is composed by a genome enveloped by a protein capsid, and enveloped viruses have a lipidic layer around. And considering the different agents, so for example, peroxides, halogens, aldehydes, they will more act on the genome, or the proteins. But in revenge, ammonium, the quaternary ammoniums or other tensioactive will more act on the lipidic layer. So in this case, we will remove the COVID-19 from the surfaces. So the last part is about the regularity aspect, and I let the floor to Laurent.
Okay, so for this last part, we would like to know which molecule can be considered as a virucide. So let's focus on the European regulation. So a virucide is included in the European biocide regulation and have to meet multiple requirements. One of the requirements is to perform efficacy tests on several enveloped and non-enveloped viruses according to strict international standards and protocols. So a close list of active substances has been established, and only biocide comprising one or more active substances listed at the European level may be sold on the European market. But the European legislation is still in transition regime. So, for example, some molecule can be accepted at the national level, but not at the European level. So it's a very complex matter. So simply ask to your provider to provide you your virucidal certificate according to your use and to your risk. Okay? So a take-home message. So in order to ensure good and cleaning disinfection practice, it is very important, first, to respect the protocols such as contact time and application temperature, to select the products that will be effective according to your environment, your activity, and of course your risk, and to use the right method for the application. And more specifically for the risk COVID-19. This type of virus is very fragile due to the presence of its lipid membrane. It may be present on multiple surfaces and may be in biofilms. It is eliminated effectively by choosing the right molecules. So before, before to finish, some words about Realco. So Realco is located in Belgium in the Science Park of Louvain University, and our objective is to reinvent hygiene for the well-being of humans and for the environment. Our vision is to be the leader in enzymatic detergents, biofilm treatment, and their prevention. We are currently 60 people, including 7 people in the R&D team, and we have strong collaboration with university and research center in Europe and across the world. We have currently 30 patents on enzymatic detergent and their application. We are members of working groups about cleaning and hygiene in the food industry. And one of our key successes is to continue to innovate with 15% of our turnover reinvested each year in the R&D. So thank you for your attention, and do not hesitate if you have any question. We have a chat box, and you can pose your question directly in the chat box, and we can give you a response directly. Thank you again. Okay. Yes, there is a question about, can we email this presentation? Yes, of course, we have all the information and you can find this presentation in the next few days. So, Biocides certificate, is it obligatory? Yes, if you want, if you have this risk in your industry, you have to provide this certificate in order to ensure your disinfection. It depends of your risk, of course. So if you are sure that you have this specific risk in your industry, please ask to your provider this certificate. Yes. So is your agent legalized in the Netherlands or internal European level? Yes, we have. We can provide our products across Europe through our distribution partners. So if you want to have our product, you can contact us directly, and we can give you the name of your partner. Okay, still one, yes. So yes, about US and South America. So in the US, we have a company. This company is Lysianne, and we can sell directly our product in the US market. For the South America, we begin to develop our business in this part of the world, but it's not yet strongly developed. So it's easier to have our product in the US than the South America. Yes, I don't understand this question. Is that spelled resign for US? No, it's a real Zyme. We will write the name of a company in the chat box. Okay. So, okay, but it seems that there is no more questions. So thank you again to be here today for this webinar. And we hope that all the information useful for you and your application. So if you have more questions, do not hesitate to send us an email or to contact us. It will be a pleasure for us to give you more information about our product or general information about cleaning and disinfection, and in particularly on specific risk. So thank you again, and see you in the next webinar next week. Thank you again.